I am Valentina Mazzotta, physician in the National Institute of Infectious Disease Lazzaro Spallanzani in Rome, and I work in the unit of HIV, Mpox, and SARS-CoV-2. Recent data of in vitro neutralization showed a reduction of the activity of some monoclonal antibodies against sublineages of Omicron SARS-CoV-2. But the impact of this in vitro data on the in vivo efficacy of the treatment are still unclear. Our study was a randomized trial, phase 4, open label, with the aim to compare different treatments of early SARS-CoV-2 patients with mild to moderate COVID-19 and who are at high risk for progression were randomized to receive either sotrovimab intravenous or tixagevimab-silgavimab intramuscular or nirmatrelvir ritonavir orally. They were evaluated at baseline and after 7 days and after 29 days from randomization. At each visit, they collected a nasopharyngeal swab for the measurement of viral load expressed in CT cycle threshold. And the primary endpoint of the study was to compare clinical efficacy assessed as a composite endpoint, which included death, hospitalization for any cause, or progression to severe COVID-19. The secondary endpoint was to assess the variation in viral load in the nasopharyngeal swab from the randomization to day 7. Among the 400 patients that we analyzed for the primary endpoints, we demonstrate a very low rate of clinical failure, that was 1.25%. We observed no death and 5 hospitalizations, 1 in the sotrovimab arm, 4 in the tixagevimab-silgavimab arm, and 0 in the nirmatrelvir ritonavir arm. The pairwise comparison showed that patients treated with nirmatrelvir ritonavir may have an advantage over those treated with tixagevimab-silgavimab, but this analysis is inconclusive for a superiority of this treatment because of the low number of events. Regarding the secondary endpoint, the viral load decrease from randomization to day 7, we observed that at the enrollment, viral load was similar across the 3 arms, but at day 7 we observed a strong activity, a stronger antiviral activity of nirmatrelvir ritonavir in decreasing viral load. The viral load variation was used in phase 2 clinical trial of some monoclonal antibodies, and actually in clinical practice is a common test to measure viral load in the nasopharyngeal swab. We know that the reproducibility of the viral load in nasopharyngeal swab is not so feasible as, for example, in other kind of tests as sample as blood with viremia. But in this Omicron era, due to the lower probability to be hospitalized and the highly percentage of persons vaccinated, probably clinical trial need other endpoint different from the classical endpoint as hospitalization and death that were used in phase 3 clinical trials. So we propose this viral load measurement, viral load change, as a surrogate marker of viral activity for assessment of drugs efficacy. Okay, actually, nirmatrelvir, ritonavir are the drug more used because, for example, in Italy, general practitioner can prescribe nirmatrelvir, ritonavir, so it's widely used, and guidelines recommend it. But there is some limitation, for example, in the population who can benefit of this drug. So researchers and clinicians need to investigate further for mechanism of action of other drugs in order to tailor the treatment of COVID-19 for patients that cannot benefit from the antiviral nirmatrelvir, ritonavir.